Okay. Now yesterday we have looked at, means day before yesterday we looked at one of the uh, protecting tissue, that is epidermis. Now today, so we will look into another important tissue, that is cork. Cork is also considered as one of the uh, protective tissue. Cork generally observed in old stems, and this is in general concentrated more in the part of stem. Are you able to see the screen? My screen?
Yes, sir.
Is my voice is audible?
Yes, sir. Yes, sir.
Okay. So now, uh, cork is one of the protective tissue, and I said it is present in older stems, and often you may see in old roots also. So these are the two locations where we see cork as a protective tissue. Okay. Now, uh, how this cork is formed? We all know that there are three types of meristematic tissue. Among three, which one of the meristem will help in the increasing the girth of the stem? Can anyone say?
Lateral.
Very good, Pallai. So it is the lateral meristem which helps in the increasing and the girth. In the sense, it is nothing but it helps in the plant secondary growth. So secondary growth is under the control of lateral meristems, and these meristems divide and redivide, produce new tissue in the stem due to the girth of the stem increases, the diameter of the stem increases. So that is what we call it the secondary growth. Now the cork is the result of secondary growth. Cork formation, so you all please make a note of the cork is result of secondary growth. You are able to see the diagram above. The outermost layer you are seeing, this outermost layer is in general nothing but so this ruptured cells are nothing but initially the stem and root had epidermis. This epidermis is ruptured during the secondary growth. Because the secondary growth damaged the epidermis, there should be another tissue which should protect the internal structures of root and stem. So that's why the cork has evolved during the secondary growth as one of the protective tissue. Okay. Now how this cork is formed? Cork is generally formed from cortex region. You can see cork is formed from a structure called Carcambium. See, I will see this Carcambium is generally formed in cortex of the stem and root. I can say, for your knowledge, you just only remember these terminologies so that they can be easily memorized and presented in the examination, because we are not need to go to the deeper parts of how this Carcambium forms, what is all these things. Now just I will make you clear regarding, suppose I'm just drawing uh, okay. Okay. Now here, this is the transfer section of stem, you imagine. So inside this, at the middle of the stem or root, you will see complex tissues like xylem and phloem. And this outermost layer, which we call epidermis, we call it as epidermis. Now, the region which is present in between the epidermis and the vascular tissue. Vascular tissue means where xylem and phloem are present in the root and stem. This region is called cortex. This region is called cortex. Now, some of the cells in the cortex region, so like I am drawing, these are the cells in the cortex region, which form Carcambium. Now, these cells are formed when there is a secondary growth. As such, Carcambium is not present in the cortex. Some of the cortical cells will form the Carcambium. So, cortex is made up of parenchyma. Cortex is made up of parenchyma tissue. Some of the parenchyma cells will form Carcambium during the secondary growth. This happens only during secondary growth. Every time you don't see Carcambium in the cortex. Secondary growth. So now suppose if the Carcambium is formed during the secondary growth, you say. Now what happens to the Carcambium? I am just giving you the nature of Carcambium in the form of a linear cell. So you imagine these blue stripes which I am drawing as a Carcambium. Now, so like this, you will see entire circular strip of Carcambium inside the cortex of stem and root. Now what happens? This Carcambium divides periclinally. Periclinally means the division parallel to the ground. So now let us divide. So now this cell has divided, 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 this cell has divided. Okay. Now these outside cells, they form cork. The inside cell, they form secondary cortex. So outside cells from the Carcambium, the blue one is called Carcambium. And now this Carcambium divides periclinally, that is parallel to the ground surface or parallel to the surface, and it forms two layers of cells. The upper layer forms cortex and the lower layer forms, no, the upper layer forms, cortex cork tissue, and the lower layer forms secondary cortex. Why we call it as a secondary cortex? Already cortex is present inside that. So the newly formed cortex we call it as a secondary cortex. The already existing cortex we call primary cortex. So in this way, by the division of Carcambium in the cortex region of the stem and root, periclinally, the upper side it forms cork and the lower side it forms secondary cortex. That is how the cork is formed. That's what we have seen in this diagram. All these are cells which are formed from the division of Carcambium towards upper side. You will see several layers of such cork tissue is formed. The cork is technically called phylum. You just remember. The technical term which we use for the cork cell is phylum. Okay. The technical term which we use for the secondary cortex, the technical which we use for secondary phylloderm. The technical term for the secondary cortex is called phylloderm. The technical term for the cork is called phylum. And similarly, the technical term for the Carcambium is called phylloderm. Not phylloderm, phyllogen. So phyllogen means generator of phylum. That is general formation of cortex. Carcambium divides periclinally, and the outer side it forms phylum, and towards inner side it forms phylloderm. So these are the three terminologies that you should remember. So they may ask for 1 mark questions. What is the technical term for cork? What is the technical term for secondary cortex? And what is technical term for Carcambium? So these are all the things which are in general seen. Now coming to the cork cells structure. Let us describe how these cork cells are present. Now when we look at the cork cells are basically, they are more or less rectangle in shape. They are having rectangle in nature. So you can see this is more or less rectangle in shape. Okay. Now they arrange very compactly, means the cells have seen very compactly, they are arranged without any gaps. And they are not one single row. You have several rows of such compactly arranged cork cells. They are present in several layers, and you don't find intercellular spaces. Nowhere the cell has left intercellular spaces. All the cells are very tightly packed without leaving any gap between the adjacent cells, which we generally consider it as there is no intercellular spaces. There are no intercellular spaces that we see in this. There are no intercellular spaces, and they are rectangle in shape. They are present in several layers. Now, the old cells of corks are considered as a dead cells, and the dead cork cells are basically gives space to fill some unwanted waste material of the plant, like we can say resins. So you might be heard this resins, gum, tannins. So these are the substances which are waste material to the plant, and these waste materials are stored in the cortex. Outer cork means outer cork cells. These substances are stored because the outer cork cells are dead cells. So that's why these are all stored in this. Now, the wall of the cork is in general made up of a substance called suberin. So you have to. So this is the substance which makes wall of the cork. Wall of the cork is in general made up of a substance called suberin. Okay. Now when it is tightly packed, there is no means the gases cannot be get exchanged in the case of stems and roots. So in order to allow the the exchange of gases, some of the regions this cork modify and forms a special pores or loosely arranged structure called lenticel. So lenticels are in general specially loosely arranged cork tissue region it meant for the exchange of gases, and these lenticels are only present in old stem, not even in the root. Only in old stems you will see lenticels. So let me show you how a lenticel looks. Uh, Now on the stem you are able to see these kind structures. All these structures, what we can say, they are of lenticels only. What I can say. So this is. So on the bark of the stem, you will see these patches. Now you are able to see these structures. So these structures which are formed on the old stem are nothing but they are the lenticels. These are these are formed by the loosely arrangement of cork tissue which helps the stem for the exchange of gases. Now let me tell you one thing. If it is in a different form, let us see. So this is the structure which is showing about how internally you will see the lenticels. This is the bark which is result of secondary growth. Now this structure is in general we consider it as a, see, whatever the terms I have used, every term is present here. So this is what we call it as a phylum cork, and this is called Carcambium. So internally Carcambium produces phylloderm which is called secondary cortex. Externally it forms cork. So phylum, phyllogen and phylloderm. Now here you are able to see the cork is loosely arranged. See here you can see very compact. This is very tight and this is very tight without any intercellular spaces. But in some regions in between this, it has very loosely arranged cells. So this loosely arranged tissue which is seen on the old stems, which is mainly meant for the exchange of gases, we call it lenticels. So do you understood what is lenticel?
Yes, sir.
Okay, this is what lenticel and how it looks internally. How it looks internally when you cut the stem, it looks like this. If you see directly on the stem, they are like this. See, these are the things which we see. These are the structures which we call as lenticels. So they are mainly meant for the exchange of gases. This only you can see in the old stems. Okay. Now let me come to the functions of cork. When we talk about the functions, uh, the first function is protection against water loss. So it doesn't allow the water to lose from the plant parts. So first priority is the water which is present in the plant is protected against water evaporation. It doesn't allow the evaporation of water. Secondly, it helps in protection from microorganisms. So protection from microbes is another important feature of the cork, which doesn't allow the microbes to get into the plant. It prevents the entry of microbes. And secondly, it acts as insulatory organ. It acts as insulation. In the sense, it protects the plant from high temperature. Similarly, browsing animals, browsing or grazing animals. And thirdly, we can say that it protects the internal structures from injury. So these are all some of the uh, properties which comes under insulation. And next property of cork is to form lenticels. Just now we have discussed. So lenticels are the product of cork only, which are in general meant for the exchange of gases. Exchange of gases. And along with this, the cork is having a commercial importance. Cork is having commercial importance. Basically, it is commercially used in stoppers. So you can see stoppers for the bottle. All the bottle caps will have at the bottom, which is made up of cork. It also acts as a shock absorber. Cork is used to absorb the shocks, and it is used widely in sports, goods preparation. Cork ball, you are all aware of cork ball. So cork ball is entirely made up of cork tissue and other uh, different types of materials. Widely this cork is used. So this is how normally regarding the cork. Now let us, with this we have finished the protective tissues. Now let us move to the complex tissues now. So as I have already told you, complex tissues are basically what we can say that uh, can anyone define what is complex tissue? Because I have already told you the definition of complex tissue. What is,
Made from different types of cells.
okay, then, okay, let me, you have given one distinguished feature. One, first of all, complex tissue is a permanent tissue. It is a permanent tissue which is in general made up of more than one type of cell. Made up of more than one type. Their origin is different. their origin is different. Means, suppose you imagine there are four types of cells, more than one. I just I'm giving you an example, four types of cells. All these four types of cells are formed from four different mother initials. Okay, so that's why all the types of cells are formed from different initials. So that's why. But all these different cells which are present in this permanent tissue are in general work together to perform a particular function. Means all these work together for particular function. So that's why we call it as complex tissue. Because so far we have seen the simple tissues where they are made up of a group of cells which are having similar in their origin, similar in their structures, and all together they perform a function. Now, when we come to the complex tissues are entirely different. They are made up of more than one type of cell, and all these cells are formed from different initials, even though their structure is different, but all together they work for a particular function. That is what we call it as a complex tissue. Now, in plants, we have two such complex tissues are present. Means we have two types of complex tissues. So these two tissues, so one is called xylem, the other one is called as phloem. Now, these are together called as even vascular tissue. Xylem and phloem are called together as vascular tissue because they are located in majority vascular bundle. And they deal with the function of water conduction by xylem and food translocation by phloem. Okay, water conduction by xylem and food conduction by phloem. So this is how normally we call these tissues as complex tissues. We may also call it as a vascular tissues because they both are involving in transportation of water and food material. That's why they are also called conducting tissues. They are also called as a conducting tissues. So let today look into the one of the conducting tissue, that is xylem. Now the meaning of xylon means wood. Means the xylem contributes the major portion of the wood in the stem. The wood of the stem is made up of xylem. So that is meaning of xylem. So when we deal with the xylem, so we need to know that this is a complex permanent tissue. First point we need to note is it is a complex permanent tissue. Okay. Next, it take part in conduction of water and minerals. So it take part in the conduction of water and minerals inside the plant. Now, if you look into the other properties, xylem is also provides mechanical support. Along with the above functions, it also provides plant mechanical support. Now, what are the different types of cells present in the xylem? First of all, let me talk. There are four types of cells present in. Number one, xylem consists of four types of cells, or you can also call elements. Instead of cells, we can also call elements. It made up of four types of elements. They are one, tracheids, and number two, vessels. Number three, xylem parenchyma. So in the xylem, parenchyma tissue is also present. So that's why we call such that parenchyma as xylem parenchyma. Fourth one, xylem consists of sclerenchyma also. So it xylem sclerenchyma. So as I have told you, in addition to the conduction of water, it has provides mechanical support to the plant. It is because of the presence of sclerenchyma only. So the sclerenchyma which is present in the xylem tissue, we call it as a xylem sclerenchyma. Now here 1 and 2, these are in general tracheids and vessels are in general nothing but tubes for conduction of water and minerals. And along with these, parenchyma and sclerenchyma are also present in the case of. Now let us see how these tracheids, vessels and xylem parenchyma, sclerenchyma look. Now first let us talk about tracheids. Basically, so this is the structure. What I have drawn is the structure of tracheid. This is how looks the tracheid is. So on the walls of the tracheids, you will have minute pores, which we call pit. Okay, this is what we call tracheid. Now, so it's a very long tubular cell, and it is a dead cell. It is a dead cell. It has thickened with lignin substance. So that's why it has lignified walls. Because of the presence of lignin in the walls, it is called lignified walls. And it has a lumen but tapered at the ends. So if you see the ends, you will see they are like tapered ends, we can say. So it is long tubular dead cell with lignified walls, wide lumen and tapering ends are present. That is how the structure you can describe the tracheids. So here you can see here and there we have seen there are small pits are present. That is also one of the character. So tracheids are water conducting elements in lower plants. In higher plants, the number of tracheids are initially present in primary structure, but they are later replaced by high capacity tubular structures which are called as vessels. So this is in general regarding the tracheids. Now we will look into vessel. Vessel is nothing but a bigger tube to carry more amount of water. So the tracheid helps to conduct more amount of water when it is compared to the uh, tracheid. Vessel is a bigger tube compared to the vessel. So vessel is also called technically trachea. So trachea is a technical term for vessel. So normally if you see the vessel is in general, it's a broad when we compare with the here also you will see small minute pores which we call them as pits. This is how normally we can see. Now they are very long. Tracheids are first of all they are very long, which are formed by the end-to-end union. Means here you will see one more vessel is present in the bottom and you are in the top one more vessel is formed. So like this it forms the entire complete structure of the vessel. Here even vessel is also a dead element. The transverse walls of these vessels contain pits which are characteristics and they also made up of lignin. So the vessels are also having same lignified walls. So when we look at the vessel and tracheid, their purpose is same to transport or what we can say that conduct water. But the amount of water they are conducted are different. Tracheids are taken away or conduct less amount of water. The capacity of uh, carrying more amount of water is high in the case of vessels. So that's why in higher plants you will see the presence of vessels and in the lower plants you will see the presence of means majority tracheids are present in the plants like pteridophytes and gymnosperms. So these are the two groups of plants in which tracheids are present as a conducting element of water and minerals. But the vessel is present in angiosperms. So this is a characteristic feature of angiosperm. Presence of vessel in the xylem is a characteristic feature of angiosperms and presence of tracheid in the xylem is a characteristic feature of pteridophytes and gymnosperms. So this is the structure regarding the xylem, uh, tracheid as well as uh, vessel that is trachea. Now coming to the other two components which are present in the xylem are xylem sclerenchyma. Now this xylem sclerenchyma is also called xylem fibers. So the other name for the sclerenchyma in the xylem is xylem fibers. As we all know that the sclerenchyma consists of two types of cells. One are long fibrous cells and short scleroid cells. So only long cells are present in the xylem. So that's why we call these sclerenchyma as xylem fibers. So now The xylem looks like similar where it is present in the sclerenchyma fibrous. So it is having a smaller, narrow lumen with a thickened wall, which is also thick with the lignified thickening, and it has a narrow lumen. The similar way the structure, so what we have seen in the sclerenchyma fiber cells, the same structure it is maintained even in the fibers also. So like this you can see. Now here the lumen is narrow, ends are tapered. It also will have small pits. Pits are present even in the walls of the xylem also. So the main function of the xylem fibers in the xylem is it is to provide mechanical support to the tissue. It is to provide mechanical support to the tissue. Now the last one which is left out is Xylem Parenchyma. The parenchyma which is present in the xylem, we call it as a Xylem Parenchyma, and this is the only living cell in the xylem tissue. It is the only living cell in the xylem tissue. Other three cells like tracheids, vessels, and even sclerenchyma is also a dead. We have already studied regarding the structure of sclerenchyma. It is a dead mechanical tissue. So the one which is only present in the xylem, which is a living, is xylem parenchyma. So xylem parenchyma cells are basically, they resemble parenchyma cells. So they are in general, they have a rectangular cells which has a nucleus and the cytoplasm, and all these normal cells are in general like you will see parenchyma cells are present like this in the xylem. So the main function of the parenchyma in the xylem, it has one, store food material, as well as it also involved in the lateral conduction of water. Lateral conduction, both are the functions of the xylem parenchyma. As a whole, when we talk about the functions of xylem, as a whole, when we talk about the xylem function, one, it is in general helps in conduction of sap. Conduction of sap in the sense, xylem conducts water and minerals. So from the roots to the aerial parts of the plant. The movement of water and minerals are in general, they are unidirectional. Unidirectional in the sense they are transported or conducted from the roots to the aerial parts of the plant. This is first and foremost important function of xylem. And secondly, it provides mechanical support. So mechanical support is another function of the xylem. Xylem provides mechanical strength to the plant because of occurrence of thick walled lignified components inside it, and xylem is the source of wood. Xylem is the source of wood. It is in general, we also, the wood of the plant is mainly made up of xylem, which has a commercial important role for the human. So with this, I am ending the today's session on tissues, plant tissues. We have completed cork, which is comes under simple protective tissue. Now today we have taken up complex tissues, and we have finished the first part of the complex tissue, that is xylem, and which is very important for the examination point of view. So I urge all of you, please go through this video even after posting into your groups, And if you have any doubt, right now you can ask if you have any doubt regarding whatever we have so far covered today's topic. And you will be having class on Tuesday, not Monday. Tuesday, same time, 8 to 9 o'clock, Tuesday. If you have any doubt, you can ask. Otherwise, you are all free to leave the class.